Mm. Sorry for being late. It was actually my problem, and that is I had clicked on the Zoom, and normally the window pops up. And I notice now there's a start button I have to push, and I didn't push the start button until recently. Any questions before we begin? How do I get rid of that? All right, not hearing any questions. Let me figure out where did we drop off? It didn't start where we stopped last time. Boy, that's not right. One of three. No, it's not that one. Let's erase that. I don't think it's that one either. Oh, there we are. Viral multiplication bacteriophage. That's where we were. All right. Let me share
Your
my screen
screen is
here.
black.
I'm sorry.
Your screen is black.
Yeah, I know I haven't shared it.
Okay.
I've got a few buttons here. Well, that's disappeared now. All right, I have that we were talking about viral multiplication last time. We mentioned that bacteria viruses, which are called bacteriophages, can multiply by the lytic cycle or by the lysogenic cycle. Anyone disagree this is not where we started last time or ended? Oh, let me start with the syllabus. Sorry. Today is November 20th, and you've already taken the quiz. We'll do chapter 14 after chapter 13. Let me remind you, next week we have Thanksgiving, so there will be no class on Thursday. And next week, your unknown notes pages will close. That's Friday of Thanksgiving. So the unknown notes pages is closing on Friday, November 28th. It'll be the last time you can request a test. And the unknown project will be closing on November 29th. So turn in your unknown project by 11:59 p.m. Saturday, November 29th. Any questions about any of that? All right, let's move on. So viral multiplication in bacteriophages. How do new bacteriophages get made? What do we need? What components need to be made to make a new virus, a bacteriophage? Well, what does the virus need to replicate?
DNA or RNA?
Yeah, it needs to replicate its genome. For this slide, I'm just showing it as double-stranded DNA, but it needs to replicate its genome. It also has to make a minimum of the capsid proteins which go around the virus, because all viruses have capsid protein around its genome. So this is a minimum of what the bacterial virus must replicate or needs to do in order to replicate. We're going to talk about first the viral lytic life cycle. I don't think that link works. I don't think this computer can run a movie, but that's an older thing. And let's go in here. If it doesn't work, I'll just run the next one. I think the sound will work, but it won't show you the movie. Well, that looks like nothing's working. Sorry, I've got a new computer here. I just turned it in, and it's because Clark College started using Windows 11, and the old laptop I had uh, wasn't quite good enough to run Windows 11 correctly. Actually, I had no trouble with it, but the IT people said they weren't happy with it and they wanted to give me a new laptop. It's actually not as good. It's not as wide, the screen. And all right, let me go to the next movie because that one isn't seeming to play. Let's try that.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific.
Yeah, this is not showing you the movie. Where can I find that? Come on.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
All right, any questions about that movie? Let me just go back right here, someplace around here.
Bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is in.
The head and the phage actually contract. That puts pressure on what's inside the head, and that injects what's inside the head into the cell. I just wanted to point that out. Any questions about this movie? Let's go where we were about.
Up to 300 new virus particles can be
right there. This will be the next movie I show. So there are 5 stages to the lytic life cycle. First, there's attachment, then there's penetration, then there's biosynthesis, then there's maturation, and finally the last stage is release. In attachment, the phage literally attaches to the bacteria cell that it's going to infect. Penetration is opening up the bacteria cell so that the viral genome can be injected into the cell. And the phage actually uses the enzyme lysozyme to make a real small hole in the cell wall. And then the tail sheath contracts and that forces what's inside the head, meaning the genome, into the cell, much as a syringe, when you're working it, forces what's inside the head of the syringe into the patient. Any question about anything so far? All right, when the genome of the virus is injected into the host cell, the virus will then begin biosynthesis stage, and this will be production of both the bacteriophage DNA and the bacteriophage proteins, especially the proteins that make up the capsid, the protein code around the genome of the virus. And this is talking about for a DNA virus. If it's for an RNA virus, obviously you will be replicating your genome, which would be RNA. Any questions about biosynthesis? All right, the next stage is maturation. This will be assembly of the new bacteriophage particles. And what happens is the virus actually literally self-assembles, meaning the proteins start coming together and self-assemble. And before the virus is finished, there's an enzyme the virus encodes, and that enzyme actually stuffs the genome into the virus. And then the virus self-assembles, making a complete new virion. And then the virus, the last part of the genome, the last gene secreted will be for the enzyme lysozyme, which goes up on the cell wall and it digests the cell wall of the bacteria. When that happens, then water can move into the cell, and that will cause the cell to burst. Once the whole cell bursts, of course, the whole cell will die with that, and the new virions will be released. Any question about any of that? So let's talk about it in a little bit more detail here. First, the virus has to attach to its host cell. Remember that viruses are specific for what host cells they can attach to, and it has to do with the ligands or adhesions on the virus that bind to the cell receptor molecules on the host cell. And generally, most viruses are specific, attaching to, at least in animal viruses, only a few cells of one or so species. With a bacteriophage virus, it's usually one species of bacteria that can be infected. All right, and then when the virus is attached to its host cell, it will digest a very small whole right there using, once again, the enzyme lysozyme. And then the virus will, sheath will contract, and that forces the genome of the virus into the cell. Biosynthesis then occurs where the genome will be replicated, the proteins will be replicated or synthesized, whatever you want to call it, and then the virus will start self-assembling. Where do I have that? We don't have it in this one, but it's here. Maturation, where the viruses will be assembled. And like I said, there is a viral enzyme which stuffs the viral genome into the newly formed protein code. And remember that the virus can sometimes make a mistake, and instead of put in the viral genome, it can put in a piece of the host genome. That doesn't happen very often, but it does happen. Two important terms for the lytic life cycle or the burst time. This is the time from when the bacteria cell first becomes infected till the time that the cell bursts. The average is 20 to 40 minutes, depending on the viral species. and also the temperature you're growing the virus at, but averages 20 to 40 minutes. The burst size is the number of newly released virions from the host cell. The number depends on the viral species, but usually someplace between 50 and 200 new virions can be made in an infected bacteria cell. Any question about any of that? All right. The lysogenic life cycle differs from the lytic life cycle in that the host cell is not killed, it is not lysed. Upon attachment of the bacteriophage and penetration, the lysogenic virus has to decide immediately what it wants to do. It can enter the lytic life cycle and go through the lytic life cycle that I just discussed. So a lysogenic virus can enter the lytic life cycle, and it has to make this decision immediately once it infects the host cell. Any question about any of that? All right. The other thing a lysogenic virus can do, a bacteria virus can do, is it can enter the lysogenic life cycle. And that will begin the lysogenic life cycle. This cycle will continue as long as there is a protein that enforces the lysogenic life cycle. There is a repressor protein that allows the lysogenic life cycle to occur. And what this repressor protein does is it shuts down the lytic life cycle. The phage in the lysogenic life cycle remains inactive or latent. It does not synthesize the protein coat. It does not, generally, it does not replicate the viral genome. So that's why they say this virus remains latent or inactive. The bacterial host cell is termed a lysogenic cell, and it remains alive for as long as the virus remains in the lysogenic life cycle. And that life cycle is sometimes called lysogeny, and that's the same as the lysogenic life cycle. So immediately when a lysogenic virus attaches to the host cell, it has to decide, once the genome is injected into the host cell, has to decide, should it enter the lytic life cycle, in which case it goes through the lytic life cycle we just discussed, or should it enter the lysogenic life cycle? If it decides to enter the lysogenic life cycle, a couple of things happen. First, the viral genome integrates into the host cell chromosome. That viral genome is now called a prophage, where the virus is integrated into the host cell chromosome. The second thing that's important is that the a gene in the lysogenic life cycle is activated, and it's the repressor protein. The repressor protein shuts down the lytic life cycle. So none of the lytic life cycle genes are expressed. Now, when this cell goes through cellular replication and multiplication, it will copy the prophage because it's part of the chromosome. And as you know, when a cell goes through cell division, it will copy the DNA of the cell. And then we'll get two cells, each having a prophage, and both viruses will be in the lysogenic life cycle. You can go through many cell divisions where the lysogenic virus stays in the lysogenic life cycle. And so this cell has a lysogenic virus inside it. Any questions about that part? Every once in a while, for reasons which are still not entirely clear to scientists, the prophage can excise itself from the host chromosome. And then when that happens, the virus switches to the lytic life cycle. So this cell now, instead of being in the lysogenic life cycle, enters the destructive lytic life cycle. New virions will be replicated. The host cell will lyse, and that will shed the virus to another cell. And that virus will attach to the cell, inject its genome into it, and immediately this lysogenic virus has to decide, will it enter the lytic life cycle or will it enter the lysogenic life cycle? Any question about the lysogenic life cycle? All right, let's go back to the movie.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
Okay, any questions? Just a minute, there we go. Let me state there is
In a
one
way,
thing wrong
a
with
virus
this
is a
video.
parasite. The cycle does not always happen immediately. Sometimes is destroyed, and a new generation of
It's showing you bacteriophage T4. Bacteriophage T4 does not enter the lysogenic life cycle. It is a lytic virus, so it only enters the lysogenic and the lytic life cycle. Other than that, this movie is absolutely dead on. Everything is correct. Any questions about anything? All right, let's talk a little bit more about the lytic lysogenic life cycle. I've already mentioned that when the viral genome incorporates into the chromosome, we call that viral genome a prophage, viral DNA. And that prophage will express a repressor protein. While this repressor protein is active, it will keep the lytic life cycle genes dormant. So the lytic life cycle genes will not express, and the virus will not multiply in the lytic life cycle. And like I said, that can go on for many divisions where the the cell is actually living in symbiosis with the lysogenic virus and the host cell. But every once in a while, for reasons which are not fully understood, and bacteriologists, meaning the and virologists, have been studying this for over 100 years, and they're still not sure why sometimes the virus will excise itself from the chromosome and then enter the lytic life cycle. There is one event that is known to cause the virus to leave the lysogenic life cycle and then enter the lytic life cycle. And that is if you damage the DNA of this cell will leave the lysogenic life cycle and enter the lytic life cycle. It's like the virus knows that the DNA of the cell has been damaged and realizes that this cell may die, and so it enters the lytic life cycle because it knows that its copy will no longer continue if this cell dies. Any questions about any of them? All right. There are some consequences of lysogeny. First, a lysogenic cell, once it has a lysogenic virus in it, I'm talking about the bacteria cell, once it has a lysogenic virus in it, it is immune to infection by viruses of the same species of the phage, bacteriophage. Once you become infected with a lysogenic virus, you're now immune to become infected by other viruses of that species of virus. There's another consequence of lysogeny, which is much more important, and that is phage conversion, where this host cell here picks up new DNA from the virus. So this cell has new DNA and it can change the phenotype of this cell when the DNA of the virus is expressed. The best example of that is the the bacteria Corynebacterium does not produce the Corynebacterium diphtherium toxin unless the Corynebacteria diphtherium cell has a lysogenic virus in it, because it's the lysogenic virus that includes the gene for the Corynebacterium diphtherium toxin. Any question about any of that? So yes, your illness caused by a bacteria may depend on whether the bacteria itself is infected with a virus. And Corynebacterium diphtherium is much worse if it can encode the toxin for Corynebacterium diphtherium. And that only happens when the Corynebacterium diphtherium bacteria has a lysogenic virus inside it. Any question about any of that? All right, enough about bacteriophages. Let's go ahead and start talking about animal viruses. These are viruses that infect animal cells. First, the multiplication of animal viruses follows the basic pattern of bacteriophage multiplication. There are several differences, like attachment of the virus is a little different because animal cells have no cell wall. That means the animal virus has to attach to the cell membrane. Penetration or entry into the cell is also easier because there's no cell wall. You don't need the virus to contract to inject the viral genome into the cell. Penetration happens, or entry of the virus into the cell happens by one of two means: endocytosis, or by fusion of a membrane-bound virus to the cell membrane. So penetration can be different. Uncoding can be a little different, not terribly. Biosynthesis will be pretty much the same. You get production of the viral nucleic acid and production of the viral proteins. Maturation will be very similar. You'll get the nucleic acid stuffed into the virus, and then the viral capsids will self-assemble. Release can be different. You can get animal viruses which are released by rupture of the cell. So animal viruses do follow the lytic life cycle, where the virus can lyse the cell, and then that will release the newly replicated virions. But there are other ways that an animal virus can reproduce. The main way is by budding. So animal viruses can replicate by the lytic life cycle, but they can replicate by budding. And all enveloped viruses, or I should say all enveloped animal viruses, reproduce by budding. Any question about any of that? You'll note that there is no lysogeny life cycle in an animal virus. Multiplication of animal viruses follows the basic pattern of bacteriophage multiplication with a few differences. The main differences you need to know is that a bacterial virus can only replicate in the cytoplasm of a bacteria cell because that's all there is in a bacteria cell. In an animal cell, you can get the virus replicating in the cytoplasm, but the virus can also replicate in the nucleus. Any question about any of that? Another difference that you need to know is animal viruses have a different way of getting into the cell. They do have attachment that's very similar, and that is the virus has a ligand or adhesion molecule which binds to a cell receptor. So there will be certain molecules on a cell that the virus will bind to, and those are called cell receptors. And then there are certain molecules on the virus that bind specifically to the cell receptor. And depending on who the author is, those are either called ligands or adhesions. And then penetration into the virus happens by endocytosis, which you're sort of seeing that here. The virus will attach to the cell. It'll then start invaginating around the virus. And this will become a bigger invagination. Eventually, this will pinch off where the cell membrane will reform across here. And then this will pinch off, forming a vesicle around the virus. And that virus will then be inside the cell and then can move around inside the cell, inside the vesicle. But the point is, the vesicle has brought the virus into the host cell. Does anyone have an older edition of the textbook, especially edition 1 for Clark College? It might be edition 2 for Clark College as well. In the older edition of the textbooks, it was wrong and it called it penocytosis. That is not correct. The virus enters the cell by this means, and that's called endocytosis. The other way that a virus can enter the host cell is by fusion of the, let's see if I can get it here, fusion of the viral membrane. Well, that's not working right. Let's see if I can get paint here. Sorry, I'm not familiar with this computer and I don't see how to get paint. See if start does it. Oh, there we go. Up a little bit. I can. There we go. So you have a cell and it has a double membrane cell membrane, meaning a double lipid layer. And then you have a virus, and let's say this virus has a envelope around it. The envelope is a lipid bilayer. I'm not drawing that too well, but you're getting the picture. Well, the lipid bilayer of the envelope can actually fuse to the cell membrane because the lipid bilayer is mainly, I'm not getting the eraser for some reason. Oh, there we go. The lipid bilayer is mainly, oops, we need to close that off too. We can make that wider. Once the lipid bilayer of the envelope fuses with the cell membrane, you'll get something like this. Like that, like that. This coming down here. And you note that the cytoplasm here is now open to the virus, so the virus can move through, and in effect, the virus is now inside the cell. And that's how fusion of the virus envelope happens to the cell membrane. Any question about any of them? So animal virus enter by endocytosis or fusion of the viral envelope to the cell membrane, and then the new virion gets inside the cell. All right, another difference that animal viruses have, they can replicate by the lytic life cycle, so that would be very similar to a bacteriophage following the lytic life cycle, but they can also replicate by budding. When they replicate by budding, let me blow this up. What happens is the new virus will replicate, usually at low numbers, and then it will move up to the cell membrane or another membrane of the cell. That could be the nuclear membrane. It could also be the membrane of the endoplasmic reticulum. It just depends on the virus, which membrane it'll come up to. It'll come up to a membrane that has viral proteins in it, meaning you notice this virus genome and capsid protein coat is moving up to this part of the membrane, not to this part of the membrane, because this part of the membrane is lacking the proteins of the virus. And then it'll start budding on this membrane. Once again, this can be the cell membrane as shown here. It can be the nuclear membrane, or it can be the membrane of the endoplasmic reticulum. It just depends on what species of virus it is. And then it'll form a real big bud, and the viral genome will and capsid proteins will come inside the bud. And then this virus will then rip off that bud. The membrane will self-anneal, meaning it'll break right here, and then this edge will form with that edge, and that edge will form with that edge. And that's the cell membrane that's important, because you don't want the cell to necessarily die. The virus doesn't want it either, because this cell is infected, and it will, over time, reproduce more viruses. And then this membrane here also anneals, forming a membrane around the virus, and this is what we call the lipid envelope of the virus. And you can see why the membrane of a cell is similar to the membrane of a virus, and that's because that's where the virus actually obtains the envelope. Oftentimes it's the cell membrane, but it could be the nuclear membrane or the membrane of the endoplasmic reticulum. And you can see also why the virus comes up to a portion of the membrane that has viral proteins in it. And that's because when the virus wants its envelope constructed, it wants to have the viral proteins in the envelope. Any question about any of that? Anyways, that is released by budding. Here you can see budding happening. Here's a cell here. It's got 1, 2, 3, 4, 5, 6, 7, about 8 viruses budding out of it. Budding usually happens at low numbers, so that budding itself does not tend to kill the host cell. This host cell will tend to survive. If the host cell dies, it's because the immune system recognizes that this host cell is infected with the virus, and the immune system will kill this cell. So the budding virus usually does not kill the cell it buds out of. There can be an exception, and that is if too many viruses are budding out of this cell, the cell won't have enough, in this case, cell membrane or membrane of another membrane, if it's like the nuclear membrane, will not have enough of the membrane to serve the cell, and that could kill the cell. But usually a budding virus happens at low numbers, and the cell can survive the budding. So the budding virus usually does not kill the host cell. And if the host cell dies, it's because the immune system is killing it because it recognizes this cell is infected with a virus. Any question about any of that? All right. Viruses have different genomes, like double-stranded DNA, single-stranded DNA, double-strand RNA, and single-strand RNA. And remember, if you talk about single-strand RNA, you have to separate it into positive strand single-strand RNA and negative strand single-strand RNA. The virus requires slightly different mechanisms for multiplication of this genome. And then obviously biosynthesis, because it's pretty easy to, and you know how the virus will get its proteins expressed if it's got a double-stranded DNA virus. But what if it has a single-strand RNA virus, and this is a negative single-strand RNA? How does the virus express the genes of the single-strand RNA to make the proteins needed for the protein code? And anyways, my point being is that the virus will have different means for multiplication and biosynthesis depending on what its genome is. Close that down, close that down. Sorry about that, didn't see those in there. All right, any questions about any of that? If not, let's talk about DNA, viruses. There are 7 or 8 viruses. I thought I saw it 7. 7 viruses of animal cells, which are DNA viruses that cause human disease. And here on the left side, we have the family's name, and you don't really need to know about the double-strand DNA. You don't really need to know about whether it's an envelope or a naked virus either, although we have talked about that. You don't need to know about the capsid symmetry or the size, but you do have the representative genera and the diseases they cause. Like the poxviridae is the virus family that causes smallpox and cowpox. We won't talk much about it, but molluscum contagiosum also. These diseases are caused by the poxviridae. We've talked a little bit about the herpesviridae. We'll talk about them a little bit further. This includes the herpes simplex virus type 1 and herpes simplex virus type 2. Remember, that's the same species, just a different strain of the virus. Chickenpox is also a herpes viridae virus, mononucleosis, cytomegalovirus, and then roseola virus. All of these are herpes viruses. We won't really talk about papillomaviruses or polyomaviruses, mastinodonoviruses, orthohepatitis virus. We won't talk about that, although that is a virus family that includes hepatitis type B. And then the erythroviruses, the 5th disease. I thought there was one here. Oh, the adenoviruses. There's one. We will talk just a little bit about the adenoviruses because there are 3 families of human viruses that cause the human cold. And one of those 3 families is family Adenoviridae. the adenoviruses. This is the third and least most common cause of the human colds. Adenoviruses. Any question about any of that? All right. You don't need to memorize this table. I'm just pointing out there are 7 DNA families of human or animal viruses that cause human disease. For DNA containing viruses, the DNA is generally copied in the nucleus. Why does a DNA virus replicate in the nucleus? Because the nucleus of the cell has all of the parts or machinery that the virus needs to replicate its genome, which makes perfect sense because the genome is DNA, and this is where the cell replicates its DNA. There is one exception to this, and that is the pox viruses, like smallpox, is a DNA virus and it actually replicates in the cytoplasm. You don't really need to know that, but like I said, as a rule of thumb, if it's a DNA virus, it replicates in the nucleus. The only exception I know of is the pox family of viruses. Any question about any of that? And there's a herpes virus. We'll talk a little bit more about them. Once a DNA enters a host cell, one of two things can happen. First, the viral DNA can actually integrate into the host cell's chromosome, forming what's called a provirus. We don't call it a prophage because a phage is a term that's used for bacteriophages. So we call it a provirus, an animal virus that is integrated into the host cell chromosome. If this happens, the provirus remains in the host DNA permanently, meaning the viral DNA is never excised from the DNA of that cell. For as long as that cell is alive, that cell will have in its DNA the viral DNA. And the virus DNA can be replicated with the host cell DNA, in which case you'll get 2 daughter cells, each containing the viral DNA. Any question about any of that? And the second thing that can happen is the viral DNA can be expressed, producing new virions. Yeah, let's move on. And the double strand of the virus will be replicated. I'm only talking about double-stranded DNA. The genome of the virus can be replicated, and it can be the genome of the virus can be transcribed to make viral messenger RNA, to make viral proteins, including the capsid proteins. And the capsid proteins will get together with the replicated viral genome to make new progeny virions. If this virus follows the lytic life cycle, it'll lyse the cell to release the new progeny virions. The virus doesn't have to lyse the cell. It could butt out of the cell as well, and we'll talk a little bit about that in a little bit. This is showing you a virus which attaches to the host cell, which looks like a naked virus. I'm not sure if that's the case, in which case it will be brought into the cell by endocytosis. But if it has a viral envelope, the virus could be brought in by fusion of the virus envelope to the cell membrane. The virus then will be encoded. This is virus DNA, so it has to be brought into the nucleus of the cell. The viral DNA can be express to make viral messenger RNA. The viral messenger RNA will then move into the cytoplasm. The viral messenger RNA can then make capsid proteins. And here you can see the viral DNA can be replicated at the same time that the viral messenger RNA is moving into the cytoplasm. And then in this point, the viral replicated DNA will get together with the viral capsid proteins to make the new virions. And then the new virions can bud out of the cell if it follows the budding life cycle. But if it follows the lytic life cycle, the virus will lyse the cell, and that will release the newly synthesized lytic life cycle viruses. Any questions about multiplication of a DNA virus? This is showing that the viral DNA does not have to integrate into the host cell chromosome. If it does integrate, it permanently becomes a part of this cell's genome, meaning the viral DNA will never be excised from the genome of this cell for as long as that cell remains alive. All right, any questions about the DNA family of viruses? If not, let's talk about the RNA family of viruses. How do I build that up? There are 15 families of RNA viruses that cause human disease. Here are the family name in the leftmost column. You don't need to know the strand type, whether the virus is enveloped or naked. You don't need to know about the symmetry of the capsid or the size of the capsid, but you should know maybe a few of the family names and then the diseases that they cause. Family Picornaviridae is a virus you've all had before, and this virus is the #1 cause of the viruses that cause human colds. And those are actually called rhinoviruses, common human cold. We also have hepatitis A in the family Picornaviridae, and we have poliovirus in the family Picornaviridae. What do you notice about the family Picornaviridae? What's similar between polio, a common cold, and hepatitis A? Well, nobody's going to take an answer, so I guess I'll tell you there's not much similar. These are very different viruses. Polio causes a very severe paralysis. The common cold is very mild. We get this many times a year, usually. And hepatitis A can be very severe, but it's very different than polio, which is also very severe. So family Picornaviridae is a very broad family, including many different members, and they cause really different disease symptoms. Any question about any of that? All right. Like I said, you should know family Picornaviridae because it is the #1 family that causes the human colds, meaning most human colds are caused by family Picornaviridae. And because I'm talking about the human colds, let's state the virus that causes the second most common cause of the human colds, and that is Coronaviridae, family Coronaviridae. The coronaviruses were well known to scientists long before COVID-19 came around. And that's because family Coronaviridae was the second most common cause of the human colds. And then remember the third family of viruses causing the human cold is family adenovirus, which is a DNA virus. Picornaviridae and coronaviridae is an RNA family virus. We're not going to talk about calciviridae, hepiviridae, won't talk about that. Astroviridae, togaviridae. We probably should talk about some of these because these are very common causes of human diseases like rubella is a togovirus. Hepatitis C is a flavivirus, but we're not really going to talk about these. We will talk a little bit about coronaviridae, and that's mainly because this is the family of RNA viruses that cause COVID-19. Retroviridae, we'll talk about this virus. Pyromyxoviridae, I should talk about this one, but we won't. It causes very common childhood illnesses like mumps, measles. We won't talk about these. I should talk about orthomyxoviridae, but all I'm going to say is if you've never heard of orthomyxoviridae, I'll guarantee you've had it. The family orthomyxoviridae causes the flu, influenza. And we won't talk about any of these either. All right, any question about any of these? You do not need to memorize this table. You should only know maybe Picornaviridae, the number one cause of the human cold. Coronaviridae, the family that causes the human cold, is the second most common cause, and also COVID-19. We'll talk about that. Paramyxoviridae, no, I won't talk about that. Orthomyxoviridae, you should know, is the family that causes the flu. All right, here we're looking at some RNA animal viruses, and you can notice they have different shapes, different sizes. The multiplication of RNA viruses is essentially the same as DNA, with some differences because obviously it is a RNA molecule, not a DNA molecule. And I haven't talked about how DNA replicates, but you know how double-strand DNA replicates. That's the way a double-strand virus DNA molecule replicates, the same way a cell does. For an RNA virus, if it's a double-strand RNA virus, oddly enough, it does replicate very similar to the double-strand DNA, meaning what you do is you have a RNA polymerase bind to one strand of the double-strand RNA, and it just makes a complementary strand, and then both strands of the double-strand RNA virus are used as a template to make the new replicated RNA genome, and that is how a double-strand RNA virus replicates. For a single-strand RNA virus, it requires a little different method for replicating. And when we're talking about single-strand RNA, you should realize that there's a positive strand, and this is the sense strand that can be read by the ribosomes. And then there's the negative strand, single-strand RNA. This is the antisense strand, and it's complementary to the positive strand. So DNA will get replicated just by the DNA polymerase by using one strand as a template to make the other strand. And then the viral genome will get expressed as the DNA is transcribed, making viral messenger RNA. And then that'll move into the cytoplasm, and the viral messenger RNA will make viral protein. If we have a negative strand RNA genome, how it gets replicated is it makes the positive strand RNA and then uses the positive strand as a template to make more of the negative strand or the genomic strand of the virus. Remember, the negative strand RNA is never read by the ribosome to make proteins. What happens is the ribosome will use the positive strand RNA as viral messenger RNA to make the viral proteins. On the other hand, if you're a positive strand RNA virus, what you do is you make the negative strand, single strand RNA, and then use this as a template to make more of the positive strand genomic RNA. Any questions about how a RNA virus replicates its nucleus, and then how an RNA virus also makes its viral protein? It just makes the positive strand RNA genome, which could be the genomic if it's that is the genome, and then it can use that as viral messenger RNA. But if it's a negative strand RNA, it has to make the positive strand, and then that positive strand can be used as viral messenger RNA to make the viral protein. Any question about any of that? So once again, if we have negative strand RNA genome, it'll make the positive complementary strand first, and then use that positive strand to make more of the genomic negative single strand RNA. And then the positive strand RNA will be used as viral messenger RNA to make the viral proteins, which the viral proteins will then get together with the replicated viral genome to make the new progeny virions. And if it's a positive single strand RNA, you make the negative antisense strand RNA and then use that strand to make more of the genomic positive single strand RNA. And then that positive sense strand RNA can be used as viral messenger RNA to make the viral proteins. And then the viral proteins need to get together with the viral genome to make the newly formed progeny virions. Any question about any of this? So in a little more detail, we have a positive single strand RNA virus, and it will bind to the host cell by its ligands or adhesions on the virus binding to host cell receptors. This can then be brought into the cell either by endocytosis or if it has a membrane around it by fusion of the envelope to the cell membrane. The positive sense strand RNA then gets into the cytoplasm. It will then begin replicating itself, first making negative strand RNA, and then using the negative strand RNA as a template to make more of the positive genomic strand of the virion. Remember, the positive strand RNA genome can be read by the ribosome to make the viral proteins, especially the capsid proteins. And then the capsid proteins will self-assemble in maturation, taking in the viral genome, which is the positive strand here. And then the newly released virions will either be released by budding, in which case the cell will remain alive, or by lysis, which case the cell will die. And if the cell lysis, all of the new virions will be released. Budding gets released by the cell forming a bud, and then that bud breaking off to form the viral envelope, and then the membrane reforming to keep the cell alive. Any question about any of that? All right, for a negative strand, single strand RNA virus, it too will bind to those cells and enter either by fusion of the cell envelope, or excuse me, viral envelope with the cell membrane, or by endocytosis and be brought into the cell by endocytosis. The negative strand RNA gets brought into the cytoplasm and then will replicate in the cytoplasm, first making the antisense of the negative sense, which in this case will be the positive sense RNA, and then using the positive sense RNA to make more of the genomic RNA, and that's the negative single strand RNA molecule. The positive strand RNA can also be used by the ribosomes to make the viral proteins, especially the capsid proteins, and then the capsid will self-assemble with the replicated negative strand genome shown here. And then this cell will either lyse if it follows the lytic life cycle, and that will release the newly replicated virions, or it can release the virions by a bud. I haven't talked about, well, yes, I have. I talked about how a bud gets made, and that is that it simply pushes on a membrane, and the genome then moves into the pushed membrane, and then that pushed membrane will be pushed off, and that will break the bilayer of the envelope or cell membrane, and that will reform into the viral envelope. All right, any questions about how a negative antisense single-strand RNA virus multiplies? I forgot to mention, but RNA viruses, actually already mentioned it, but let me remind you that RNA viruses tend to replicate in the cytoplasm. So all members, all families of the RNA families of viruses replicate in the cytoplasm with one exception, and that's family retrovirus. This family is an RNA virus, but it does not replicate in the cytoplasm of a animal cell. It replicates in the nucleus. We'll talk about why in just a minute. All right, let's talk about family retrovirus, retroviridae. This includes the lentiviruses, including viruses that cause HIV-1 and HIV-2, both of which cause AIDS, but the symptoms are a little different between HIV-1 and HIV-2. And another lentivirus or retrovirus is feline leukemia virus, which causes leukemia in cats, and it's a sexually transmitted disease among cats. And the cat gets so sick of the leukemia that the cat's immune system begins to fail, and then the cat will get diseases very similar to human AIDS, and it's usually called kitty AIDS. But remember, a feline leukemia virus does not destroy the immune system, at least initially. It initially causes the cat to have leukemia, and then the leukemia gets so bad that the host immune system, or at least the cat's immune system, stops to function, and then the cat gets kitty AIDS. The family retroviruses are different, and they contain the gene for the enzyme reverse transcriptase. See if I can get this now. Reverse transcriptase is a protein that can take double-strand DNA. Come on, make the other strand. No, 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 no. Sorry, I'm not thinking right. What it does is it takes, come on, it takes positive single strand RNA, which is what the genome is of a family of retroviruses, meaning they're all positive single strand RNA. And reverse transcriptase, whoops, well, it's not drawn very well, but you're getting the picture. I'm just going to abbreviate that RT for reverse transcriptase. In the presence of RT, meaning reverse transcriptase, can take, let me blow that up. It can take positive single strand RNA to make single strand DNA. Put that there. Oop. And then reverse transcriptase. Let me see if I can just capture this. It's a little small, but I think you can see it. If you can't see it, let me know. If I can move that, yes, I can. The reverse transcriptase can then take single strand DNA to make double strand DNA. And this does happen in 2 steps. I think your textbook doesn't tell you that, but a reverse transcriptase has to act twice to make double strand DNA from single strand RNA. Any question about any of that? A reverse transcriptase is unique because it's the only enzyme I know that can take RNA and make DNA. Meaning it doesn't follow the law of biology that information flows from DNA to RNA to protein. In this case, information flows from RNA to DNA, single strand DNA to double strand DNA. And then this DNA can be expressed. Let's go ahead and go here. Oop, didn't get it. Sort of, it's a little small there. Pull that up a little. Oop, can't do that. Oh well, that's supposed to be an arrow. Maybe I can draw it. Kind of. It can make RNA. Which can be read by the ribosome to make viral proteins. Let's see if I can do this better. That's a little better. Get whiter. There we go. I'm just going to make this a word. V for viral proteins. That's not how you spell protein. And then these viral protein can get together with the viral messenger, the genome, make positive single strand RNA to make the new virions. I'm not sure how to do that. Any questions about how retroviruses replicate? Let me state that the double-strand DNA of a retrovirus always integrates into the host cell chromosome. So this viral DNA, which was made from the viral RNA, will integrate into the host cell chromosome, and it will permanently become a fixture of that host cell. For as long as that host cell is alive, it will contain this viral genome. Any question about anything? All right, let me come here. Oops, did I stop my share? I didn't mean to do that. I'm going to stop here. Back to here. All right, so we'll say that we're here. Any questions? If not, I'm going to end it here and we'll pick up from here next time. I will be showing up in the lab at 6:30, is that right? But I'll only be there for 15 minutes to answer questions. All right, any questions? All right,
I was
go ahead.
I was wondering if the infectious disease research project will be graded before we take our final.
It is my intention to have it graded before you take your final. I'm working on it, but obviously I've got to finish the labs, and I've also got to figure out which quiz to drop and put that number in the drop column, and then increase the points for quiz 5 up to the full amount of points. So I've got a
Okay.
number of things to do, but my goal is to get the infectious disease project done before you take the final. Hopefully I'll get that accomplished.
Okay, thank
Okay.
you.
All right. All right. If nothing else, I'm gonna say goodbye, and I'll see you in the lab if people have questions. If you don't have a question, you don't need to show up in the lab.